Hello, my name is Professor Idah Sithole Niang, the Dean of the Faculty of Science at the University of Zimbabwe. I'm here today to produce a promotional video in recognition of my appointment as a member of the Academic Union Oxford. This membership comes with an honorary professorship as well. And this is in celebration of that, I thought we'll share just briefly a few of our achievements. Our laboratory over the years has made several products through gene expression, where we express genes and make proteins because it attacks all gram-negative bacteria, so a whole group in the whole spectrum of that. Secondly, we've also made an antibiotic that is attacks methicillin-resistant Staphylococcus aureus and can be used in the treatment of mastitis in dairy cows. We've also produced a vaccine for the foot and mouth disease virus, which is a prevalent viral disease in the low-lying regions of Zimbabwe, and it affects the global disease. But we've made a vaccine out of this, and this vaccine, the virus itself, the coat, we're using protein coat. The coat is made up of 4 components, which essentially each component is immunogenic, so it's a potential vaccine. And then when it self-assembles, it also makes the vaccine. So we're awfully proud of this. And again, we have dovetailed it into light induction. So all our genes are being driven by solar, if you will. The natural light, and it's working very well in our hands. And then in addition to that, we have made some DNA standards. So these are molecular weight standards that are used to size DNA molecules as you run them through a matrix that we visualize to see what products we have. So we made a low-range DNA molecular weight standard from arbitrary sequence. So it's none of the standards that are on the market currently, so we made that. Then it's recently, and again manipulating the bacteriophage life cycle, we've been able to make a very popular molecular weight standard known as lambda Hindi 3. And so that again is working very well in our hands. So we produce that by mimicking the replication cycle and then making the standard from that. We've also produced a range of high-grade molecular biology reagents that are aimed at input substitution. This encompasses 3 different types of buffers that are used for experimentation for molecular biology. We've also then, when you load the DNA sample into a gel matrix, it needs to go and be loaded under high density. So we've made the loading die as well that goes with that. Part of other work that we've been doing is to identify endophytes from either medicinal plants or indigenous Zimbabwean trees, and within that we are able to identify some interesting bioactive compounds. Specifically, we have a whole range of endophytes that, through a potting assay that we used, demonstrated that they had drug tolerance capability, if you will. So there are protectants that are being made and identified with these endophytes. And related to that, also we are finding some very interesting antimicrobial. Some of them have very important antimicrobial properties. Again, those are products that we're going to be exploiting. And we've done all this work with a group of students who come and go, and some of them now are in advanced laboratories across the globe. And perhaps with this video promotion, some of them will be able to speak as testimony and excitement of having worked in molecular biology teaching now, which is what we call our laboratory in the Department of Biotechnology and Biochemistry. Thank you.
Good day, my name is Anna Mashonganika and I'm a junior lecturer at the University of Zimbabwe, Department of Biotechnology and Biochemistry. I did my MSc research and my undergrad research at the Molecular Biotechnology Lab under the supervision of Professor Idah Sithole Niang. During my tenure, I managed to publish a paper titled The Isolation of Bacteriophages, and further to add on to that, I managed to carry out a research on the development of bacteria of Hint3 lambda DNA ladder. My experience in the lab not only fostered my passion for molecular biology, but also provided an environment whereby it honed my technical skills as well as professional skills in molecular biology. I would endorse this lab as a premier environment for developing young scientists to come wholeheartedly. Thank you.